Hello friend, today in this video I am going to talk about nucleophilic displacement on a phosphorus atom. phosphorus atom पर nucleophilic displacement कैसे करते हैं, ये जो टॉपिक है बायो ऑर्गेनिक केमिस्ट्री सेमेस्टर 3 का। तो आप देखें कि रिएक्शन एट फास्फोरस एटम ऑफ फॉस्फेट ईस्टर कहते हैं कि जब ऑक्सीजन से R ग्रुप लगा हुआ है, तो ये देखिए फास्फोरस के बाद ऑक्सीजन की बॉन्डिंग है और ये R है, तो इसको फॉस्फेट ईस्टर ले लेंगे। एंड एनहाइड्राइड फ्रॉम द केमिकल बेसिस फॉर मेनी ऑफ द मोस्ट फंडामेंटल प्रोसेसेस इन लिविंग सिस्टम। इस तरह की सिस्टम जो है लिविंग सिस्टम में, these reactions allow generic inheritance through the nucleic acid and the coupling of chemical energy to derive the thermodynamically favorable processes required for the constructions and maintenance of the living cell. Phosphoryl transfer chemistry is also crucial for basic metabolic pathways and the cellular signal transductions. Here we discuss the nucleophilic displacement reaction on phosphorus, which is today's topic for the discussion. Now you can see this is your nucleophile, and here is the ester oxygens. So if we nucleophile attack on this system, this portion is displaced with the nucleophile. So topic, you can see nucleophilic displacement on phosphorus atom. This reaction can proceed by either of two mechanisms which are analogous to SN1 associative and SN2 dissociation mechanism. Because this one nucleophilic unimolecular or bimolecular, in another language we call this associative and dissociative mechanism. Now, if you consider dissociative mechanism, it is the elimination addition reaction. So elimination, the formation of double bond. Addition means again formation of single bond. So unstable metaphosphate intermediate form. You can see here, this is the OR. When nucleophile attack on the system, first this ester group RO minus and nucleophile, this is the metaphosphate intermediate, and then finally you have this phosphorus and nucleophile bond and RO minus. Now associative mechanisms, it is a addition elimination reaction. Pentavalent transition state formed, and this reaction is subdivided into two types. Inline mechanism. Inline mechanism, you can see the attack occurs opposite to the living group, lead to inversion of configuration. You have nucleophile, you have this system, you have phosphorane intermediate, and then your compound phosphorus nucleophile. Another adjacent mechanisms, attack occurs on the same side as the living group, lead to retention of configurations. You can see this nucleophile on the same side, the phosphorane intermediate and the formation of protein. Now apply this on the system. The coupling of ATP cleavage to endergonic process. You have here phosphorus double bond oxygen and 3 oxygen here. Adenosine 5 monophosphate AMP. This is the ADP, adenosine 5 diphosphate, and this is the ATP, adenosine 5 triphosphate. So all cells require energy to ensure their survival and reproductions. They get the energy they need by the converting nutrients into a chemically useful form of energy. The most important form of chemical energy is adenosine triphosphate. We call it ATP. So ATP, the energy currency or coin of the cell, transfers energy from chemical bond to endergonic, means energy absorbing reactions within the cell. You know, structurally ATP consists of the adenine nucleotide, ribose sugar, adenine base and phosphate group PO4 2 plus two other phosphate group. Energy is stored in the covalent bond between phosphate. You can see here, this is your adenosine and you have the triphosphate, and the energy is stored in phosphate oxygen bonds. So if you do the hydrolysis, then one phosphate is removed from the system and now it becomes ADP, adenosine diphosphate, and it gives the energy. From where this energy is coming? The breaking of phosphorus and oxygen bond. Because the hydrolysis of phosphoanhydride bond is highly exergonic reactions. Phosphoanhydride bonds are called high energy bond. The term high energy in this context means that a lot of energy is released when a reaction occurs and that causes the bond to break. The energy released after hydrolysis of ATP converts endergonic reactions into exergonic reactions. For example, the reactions of D-glucose with hydrogen phosphate to form D-glucose 6-phosphate is endergonic reaction. The hydrolysis of ATP, on the other hand, is highly exergonic reactions. You know, when the two reactions are added together, the net reaction becomes exergonic. The energy released from the hydrolysis of ATP is more than enough to drive the phosphorylation of D-glucose. So, two reactions in which the energy of one is used to drive the other are known as coupled reactions. So, this is a special type of reactions, coupled reaction. Coupled reactions means two reactions occurs with the help of each other. Now you can understand coupled reaction with this example. You can see D-glucose and hydrogen phosphate, it gives D-glucose 6-phosphate and plus water. In this case, plus 3.3 kilocalorie per mole. And on the other hand, ATP, it take this water and form ADP and release 7.3 kilocalorie per mole. So what will happen here? ATP is doing another job and the reaction occurs. Now you can see here D-glucose converted into D-glucose 6-phosphate, and the same time ATP is converted into ADP. So this is the coupling reactions. Now essentially it is an SN2 reaction with an adenosine pyrophosphate leaving group. You can see D-glucose and D-glucose 6-phosphate. So if you have the phosphoanhydride bond, adenosine, ATP, and if you have the ADP, D-glucose 6-phosphate. So in this case, you can see the enzyme catalyzed carboxylation and decarboxylation occurs. So how carboxylation and decarboxylation occurs? So decarboxylating and carboxylating enzymes have responsible for both the formations of CO2 form organic substrate and for the incorporation of the carbon dioxide in the molecule. So list of decarboxylating and carboxylating enzyme. You can see the oxaloacetate. Oxaloacetate, it gives the pyruvate by removing CO2, and with the help of oxaloacetate carboxylase, that is the enzyme. Pyruvate converted into acetaldehyde with the help of pyruvate carboxylase enzymes, and amino acid carboxylase enzyme doing conversion of this compound to this NH2 groups. Ribulose diphosphate is converted into T3 2,3-diphosphoryl glyceric acid with the help of carboxylating enzymes. So friends, I hope this material is useful for you for the examinations. Some important concept here. I will make also one video in Hindi so that most of the topic and most of the concept will be clear to you. Thanks for watching.